Hello students, today we will look into the topic on genetic engineering. This technique, this chapter basically aims to introduce you to the basic principles and applications of primarily three main kinds of genetic techniques. It's called recombinant DNA technology, polymerase chain reaction, and blotting techniques. However, this particular topic also aims to convey the importance of genetic engineering in today's medical practice. All right, let's get it started. Well, genetic engineering was very recently introduced into the medical practice. Considering the time scale in which medicine was practiced over 5,000 years. Now, why was this of interest? Now, as you know, genes are the primary source of all the entire protein product. Understanding and analyzing this gene was always a challenge because of its immense size and complexity. About 3 billion base pairs that encodes for 20,000 to 25,000 protein coding genes are located in the 23 chromosomes that you know exists. Now, in this immense complex condition, it is now possible to determine the nucleotide sequence of a large DNA, and that is through genetic engineering. Now, it is all made possible because we were able to develop methods that can manipulate the DNA cell to produce various biological product and therefore change the hereditary trait. We could also determine the protein expression of these genes in terms of protein production. Now, this entire project was termed genome project or human genome project, wherein a bunch of people got together to sequence the entire genome. It took quite a heck of a time and a lot of money and resources, but it was done somewhere in early 2000s. Now, this technique, genetic technique, is currently used for various reasons, which includes identifying various genetic disorders and gene therapy, where in gene therapy is, although at a very infantile stage, it still holds a lot of promise. What basically gene therapy means is that it manipulates the defective gene and introduces the correct gene sequence wherever there is a defect identified. Well, here are some common concepts and techniques that you should be familiar before you go on to learn on these three main genetic techniques. Cloning. Cloning is used to produce an exact replica of a particular DNA or a strand of a DNA or a fragment of a DNA that you're going to use or for protein production or genetic identification. Genetic probes. These includes genetic probes means a dye that is used to identify a certain sequence of DNA fragment. We will learn about this in the later slides. Recombinant DNA. Now, what is a recombinant DNA? Well, this is a DNA that has a combination of DNA from two different sources to create a new DNA called a recombinant DNA. Restriction endonuclease. This is a fantastic enzyme that allows this recombinant DNA to be manufactured. More about it in the coming slides. Gene therapy. It's the ability or the method that is adopted to modify DNA of an organism to repair defects or create a desired change in the genome. Now, there are various techniques that exist in the realm of genetic engineering, which includes recombinant DNA, polymerase chain reaction, blotting techniques, DNA sequencing, also called karyotyping, DNA microarray, protein engineering, hybridoma technology, mutation detection techniques. Now, we are interested in the top four. Let's look at recombinant DNA technology to begin with. Well, recombinant DNA technology, the conceptualization and the methodology was given by Boyer and Cohen way back in 1973. It basically involves the following principle. What you do is you take genome of interest which you want to multiply or produce a large number of a specific DNA fragment, let's say from a human source. For example, let's take insulin. You need to produce insulin protein, and that insulin protein is there in a gene in humans. So you take that particular fragment of a gene coding for insulin. You take this gene fragment and combine it with a gene of another source. Let's say, for example, a prokaryote. It can be a bacteria. So you incorporate this human gene into the bacterial gene to form a chimeric DNA. And this is done by this enzyme restriction endonuclease. Once this chimeric DNA is manufactured, you introduce this chimeric DNA into a vector, in this case, possibly a vehicle or an organism which is capable of multiplying itself very rapidly in a short span of time. Now, incorporation of or preparation of chimeric DNA also includes another enzyme called DNA ligase. Now, once the DNA of interest is introduced into the vector, the entire chimeric DNA undergoes containing bacteria undergoes cloning, and there is an expression of this particular desired gene of interest into many folds. So basically, if I have to express it in this particular picture, it appears something like this. Now, this is the DNA of a human cell. So suppose you need to take the insulin gene which is existing in this particular portion of a human DNA. What you do is you take the gene and identify the specific chromosome, and from that specific chromosome you have various techniques to identify this particular sequence. And once you identify this particular sequence, you cut it by using this enzyme called restriction endonuclease. This is a very important enzyme. We will talk about it in the next slide. So what this enzyme does is it cuts the entire genome into small fragments. And what we do is we use various probes to identify the insulin gene containing fragment. Once we do that, we go to the bacterium now. We go to the bacterium, we use the same restriction endonuclease to cleave what we call the bacterial plasmid. Now, plasmid is a different set of prokaryotic genome, which is different from the genome with which the bacterium multiplies. This plasmid contains genome in which bacteria learns to thrive or to react to an external environment. I think you will learn more about bacterial plasmids in your microbiology class. For today's explanation, just assume that bacterial plasmids is another set of circular DNA that is present inside the bacterium. So once we use this restriction enzyme, the restriction enzyme cuts the bacterial DNA also. So now you have the fragment, now you have the bacterial DNA, both are combined, and when you combine it, you get this recombinant DNA. Now this DNA is introduced into another vector for it to be multiplied. Okay. What is this interesting restriction endonuclease enzyme? Now, what this restriction enzyme, endonuclease enzyme is, it is an enzyme that has an ability to recognize short stretches of double-stranded DNA that contains specific nucleotide sequence. Now, it is also used to cut single-stranded DNA, so it does cut both double-stranded and single-stranded DNA. Now, because each enzyme cleaves DNA at a specific nucleotide sequence, restriction enzymes are used to prepare various lengths of restriction fragments. All right, just to give you an example, this restriction enzyme has a very unique characteristic sequence which it can identify, and that's called the palindromic sequence. What's a palindromic sequence? Well, look at this example over here. Now, this is an example where you have a GAATTC in one of the strands of the double-stranded DNA, and then in the opposite strand you have CTTAAG. So this is a long sequence, and look at this other sequence, AATT, and the other template has got TTAA. Now, the interesting thing about this sequence is when you read this sequence in one direction and look at the other sequence, it reads the same in the opposite direction. Such a sequence is called palindromic sequence. 5' to 3' and 3' to 5' sequence is the same. So restriction endonuclease of various sources recognizes various types of palindromic sequence. Now, this sequence is present across the entire genome at different spots or spaces. Wherever the sequence is there, this restriction endonuclease comes into the picture and cuts, and it can cut the DNA. It can cut this particular sequence and separate the two strands. So this cut produces either a blunt end or a sticky end. What do I mean by blunt end? Imagine this to be the DNA that needs to be cut. Now, this particular cut, this DNA has to be cut, and it can cut in two ways. One is through blunt end. So when it cuts, the cut is straight. This is a straight cut, so the edges are absolutely blunt. Now, what do I mean by blunt? You'll appreciate it when you look at the sticky ends. Now, the same, there can be another restriction enzyme which can cut in a staggered fashion. When it cuts the staggered fashion, a small portion of this particular DNA is got a nucleotide sequence which is exposed, and another small portion over here gets exposed. So this is called the sticky ends. It makes it easier when the bacteria has got sticky ends, or the restriction endonuclease produces sticky ends to form a chimeric DNA. It's much more easier and stabler when compared to blunt ends. So blunt ends are not very effective in forming a chimeric DNA when compared to sticky ends. So what this particular restriction enzyme does is it recognizes these restriction sites and cuts the entire fragment of DNA into different shapes, into different sizes, depending upon where it recognizes these sequences. So these are called restriction sites. Now, once these sites are recognized, now this is another example of a sticky end that is cut by this specific restriction enzyme, endonuclease enzyme. The word endonuclease, I should say, means cutting in between. That's why it's called endonuclease, not exonuclease. So this exonucleases, on the other hand, cuts it from the ends. All right, so that's why this is called restriction endonuclease. And look at the example over here of a bacteria from bacteria called Escherichia coli. So this gives a sticky end, right? Now, just to suggest to you that this particular restriction endonuclease can be obtained from different sources. Now, depending upon the source which it is obtained, the enzyme is labeled with that particular name. For example, Escherichia coli is one organism from which a restriction endonuclease can be synthesized, and that's why this is called E. coli. Similarly, Haemophilus influenza, that's why it's called HIN restriction endonuclease. All right, so the product is also given over here, whether it produces sticky end or blunt end. So you have different endonuclease to choose from already available. Now, once you have this particular fragments, how do you identify this particular the fragment of your interest? Now, this cleavage, you should understand, of human DNA produces an enormously large amount of fragments. This can be it can be a challenge to identify the specific sequence which you're looking for, and this is where you get probes. Now, what are probes? Probes are a short piece of a single strand DNA which is radio labeled. Now, this single stranded DNA which is radio labeled is the DNA which you already have an idea of what the sequence is, and this sequence is a sequence which is the complementary to the desired DNA of interest to you. So you use this DNA probe, the single stranded DNA or an RNA probe, and allow it to form a allow it to combine it with the specific sequence of the code which you want to extract. So in that sense, that particular fragment containing your gene of interest is identified using a probe. Once you identify it, you use various methods like chromatography method. This is called high performance liquid chromatography, to separate that particular fragment out and extract it out from it. Extract that particular sequence which you want to multiply or which you can use to introduce into vector. Now, vectors. Moving on to vectors. Now, what are vectors and what's the purpose and how many different types are there? Now, vector, it is a DNA which can carry the DNA of another source. It's a DNA of one organism or one prokaryote which can incorporate the DNA of another from another organism. Now, basically, there are different types of vectors depending upon how large a fragment you want to introduce. For a fragment of around 10 kilobases, 10 kilobases is the length of bases which is used, which is formed from this, which is a product of restriction endonuclease. So for a small size of 10 kilobases, you use bacterial plasmids. We know that bacterial plasmids are small circular duplex DNA, which can replicate independently within the bacteria. Now, this is one kind of a bacterial vector. Another one is called bacteriophage, which can accommodate up to 100 to 250 kilobases. And the third one is called cosmids. Now, this can accommodate between 250 to 1000 kilobases. A large fragment of a human DNA, for example, can be incorporated into this particular cosmid. What's a cosmid? It's a plasmid into which a DNA sequence from a bacteriophage has been inserted. A bacteriophage, as you all know, is a virus that infects a bacteria. So it means it carries the bacteria, the DNA, and infects it into the bacteria. So you use this to form a cosmid. Now, once the DNA of interest is added to the DNA of the bacteria, because both of these DNA has got ends which are complementary to each other, they form a hydrogen bond immediately. Now, this is what happens. Let's assume that this is the end of a human genome and this is the end of a bacterial plasmid. Now, because the same restriction endonuclease enzyme is used to cut the bacterial plasmid as well as the human DNA, these ends, the sticky ends, have got complementary base pairs and they instantly form hydrogen bonds. So it forms an effective hydrogen bonds. However, the ends over here, the ends of bacterium and the human, has to be stitched up. The ends of bacterium and human has to be stitched up over here, and this is done by this enzyme called DNA ligase. So DNA ligase's primary aim is to stitch up the ends. Once it stitches up the ends, now the entire plasmid becomes a complete circular DNA genome inside the. So the incorporation of DNA fragment to be cloned is completed with the DNA ligase and restriction and DNA ligase coming into picture. Okay, there is one key point to note over here when you talk about vectors. Now, when you're identifying a vector, you use this plasmids. Now, what plasmids do in this condition is that these plasmids have, for example, over here, they have the ability to multiply under specific condition. Each circular DNA has a genome which multiplies under specific conditions, that is in response to a specific environmental condition. For example, this is a plasmid which has got what we call is as ampicillin resistant genome. When you take a bacteria containing this plasmid or this circular DNA and expose it to ampicillin, this particular plasmid suddenly starts to clone itself. It multiplies itself. Only the plasmid multiplies. The microbe doesn't multiply. So it independently multiplies on its own. So we use such a plasmid and we incorporate the DNA into this plasmid and expose this particular bacteria to ampicillin environment, allowing the ampicillin resistant plasmid to multiply. Is this particular bacteria containing this particular plasmid, which is capable of multiplying in the presence of an ampicillin, use multiple plasmids. This is done purely because the bacteria wants to produce a lot of ampicillin resistant gene, and we take the advantage of this and we indirectly produce a lot of gene of our interest. You look over here, the number of genes that is multiplied along with the plasmid, and hence we have now numerous copies of the genome inside the plasmid. The same restriction endonuclease enzyme to again cleave this particular enzyme at the same sequence. So this in the restriction the same restriction endonuclease enzyme cleaves it at the same site, and what you get is a fragment of a DNA of your interest, which was one single fragment, has now turned into a gigantic number of fragments which can be extracted out again by HPLC methods and then later used for protein expression or study of that particular gene sequence. So this is the main purpose of recombination. So what are the other applications of DNA? First, as I've mentioned to you, is to produce a large number of the DNA of interest and to produce a protein or allow that particular genome sequence to express to produce a large amount of protein. This is a classical example of this is production of human insulin. Human insulin is a protein, it's a peptide hormone which is produced under laboratory conditions with extraction of human insulin gene. This human insulin gene is multiple copies of this is made, and these multiple copies are then later used to form multiple proteins, and this is what is used to treat insulin dependent diabetes. What are the other proteins that can be produced using DNA technology? You have anticoagulants, growth factors, erythropoietins, antibodies, and vaccines. So these are a few of the instances where recombinant DNA technology can be used to in order to produce large quantities of certain specific proteins. It is also used to understand the molecular basis of various diseases such as sickle cell anemia, thalassemia, hypercholesteremia. So we can make a large number of copies to study these genes. It's also used for gene therapy and some. All right, let's get on with the next topic. It's titled polymerase chain reaction. Now, polymerase chain reaction was a technique introduced by Klebb way back in 1971. He published this article in the Journal of Molecular Biology. Now, he introduced this particular technique after observing various bacteria and the way they react and how the genome behaves, how the double-stranded DNA behaves. And he produced a very rough skeleton of how this technique can be used to study genome. However, it was Kary Mullis in 1983 who came out with this interesting or very workable technique, which then later on caught up among the scientists working with genes. Now, this has now become a very, very popular technique. Now, what is polymerase chain reaction? Now, what polymerase chain reaction does, or the beauty of this particular technique, is it enables a large amount of DNA to be produced from a very small, tiny little sample. So the ability to make such a number, to make a large quantity is what the strength of this particular PCR technique is. It basically has a repeating cycle of two steps. One is a step which separates a double-stranded DNA. It's a one of the process of denaturation of DNA also. It's not complete denaturation. What basically happens is the double-stranded DNA is separated into two single strands. Once the two strands are separated, these two strands are now allowed to synthesize their complementary sequence of DNA. So it's like replication. It's exactly like replication. So you have two strands separated, and the two strands are allowed to make a copy of its own. Now you have four double-stranded DNA, four single-stranded DNA, or two double-stranded DNA. Again, these two double-stranded DNAs are undergo separation, and then another copy of it is formed. And in this pattern, in this repeated cycles, exponentially the number of DNAs are multiplied. Now, what are the required? What happens in this particular reaction? Now, this particular technique is so well stabilized and standardized that you have machines called PCR machines, which does the entire process of separation and formation of a new DNA strand. Now, you know that for replication process, you need a few basic ingredients. One is obviously the DNA that needs to be multiplied. Two is the primers. That means a starting portion of a small fragment of DNA to act as the starting point. The three is the nucleotides itself, the free nucleotides, your adenine, your A's, your T's, your G's, and your C's. You need all of them to be in supply. And of course, the fourth, the most important, you need all these things to come together by the enzyme DNA polymerase. So this is what is required to be fed into the PCR machine. A sample of DNA, nucleotides, DNA primers, and thermostable DNA polymerase. So basically, you are allowing a segment of DNA or a DNA to multiply itself through a process of replication. So you're mimicking replication in a laboratory condition. So we begin with first separation of a DNA. Now, separation of DNA here in this PCR technique is done by heating. So just by mere heating, the hydrogen bond holding between A's and T's and G's and C's are broken down. How much do you heat? You take the temperature up to exactly 95 degrees centigrade. This allows the two double-stranded DNA to separate out. Then what happens? You supply the primers at this particular temperature. Once you supply the primers at this particular temperature, the primer looks for the starting point, and then once it does, you reduce the temperature down to the body temperature of 37 degrees. So in this process, the primer is allowed to bind to the two separated strands of DNA. This is called renaturation. So first you denaturate and then you renaturate. This allows the primer to bind to both the starting point of DNAs of both the template and the non-template strand of DNA. And then what you do, you mix this particular, you take this mixture and you heat it again up to 72 degrees centigrade. Now why you do that is you want the process of replication to begin, and it has to begin because the DNA polymerase which you are using has got an optimum temperature of 72 degrees centigrade. Now this DNA polymerase now now reads the entire genome, manufacturing your nucleotides, your DNA of a copy of DNA of both the template and non-template strand. Once this entire genome is copied, you again go back to step 1, separation of DNA. Again you add another set of primer, again you start manufacturing the next set of DNA. So this cycle goes on, and it goes on for about 8 minutes. It takes about 8 minutes to complete one cycle for the entire genome to be copied. 8 minutes, huh? That's amazing number of nucleotides being used, and the ability of this polymerase to correctly copy them. It's fantastic. There's a small video which I would be giving links to, which you should be viewing later on after the completion of this particular presentation. Now, of course, this technique has its own unique problems, and there are solutions that have come forth. First is separation is achieved by heating at 95 degrees. Now, the problem is you are putting the entire mixture, including the enzyme restriction in the nucleus. DNA polymerase. And this DNA polymerase and the helicase, which needs to separate the DNA strand, all do not survive normally at 95 degrees centigrade. They get denatured, the protein gets denatured, and it can render the enzyme inactive. That's one major problem. To overcome that, what scientists have done over the period is to identify a specific DNA polymerase that is thermostable, or that works in a temperature between 37 to 95 degrees centigrade. See, it's important for us to take the temperature to 95 degrees. Only then will there be a separation of your two strands of DNAs. But the enzymes are also have to be maintained stably at that temperature, because these enzymes are required to form the new DNA. Now, how do we do that? We take a DNA polymerase from a source or from a bacteria which survives or thrives at that particular temperature, and that bacteria is called Thermus aquaticus. This bacteria is very commonly found underneath the sea, close to volcanic eruptions. Now, this bacteria thrives in such conditions where the temperature is very, very high. So they have taken these bacteria and they have extracted the polymerase from these bacteria, and these are called Taq polymerase. So these polymerase are used for PCR technique. So this particular polymerase has got optimum temperature working between 37 to 95, and it can be safely used in this particular technique. So once the strands are separated, the DNA polymerase comes into picture. The DNA polymerase, the problem is it cannot work with a single strand of DNA. So it needs a primer. So a primer has to be supplied. So it starts to work only in the presence of a primer and the desired set of genome to be copied. So these two together forms a double strand, for allowing the Taq polymerase to start manufacturing the DNA. What's a primer? Now, you all must be familiar with the word primer. Primer is a short sequence of DNA complementary to the base at the start of the region to be copied. Now, this was dealt in the topic on replication. So, to synthesize primers, the base sequence of the start region must be known. So, that is a given fact. So, you need to identify the primers of the genome for which you need to run the polymerase reaction. Now, here's a schematic picture or an animation which allows you to which gives you a better understanding. Now, imagine this to be a double-stranded DNA. This DNA has to be first separated, and this happens at a temperature of 95 degrees. Now, once this is separated, the primer is supplied and the temperature is brought down to 37 degrees centigrade. Now, 37 degrees centigrade allows these two primers to form a hydrogen bond at the starting point of these two genomes. Once this is done, the next step involves the Taq polymerase to coming into picture, and this Taq polymerase, which is supplied now, starts to add the nucleotide sequence, allowing the entire genome of both the template and the non-template strands to be copied. So this is the function of Taq polymerase. It uses this particular DNA double strand. As I mentioned, it needs a DNA double strand. So it only can start manufacturing if there's a double stranded DNA, and this entire genome is copied only in the presence of a double stranded DNA. This happens on both the ends. Now, if you look at it, a double stranded DNA is now replicated into two. So now there's two double-stranded DNA. So once this is formed, again you repeat the cycle. You bring the temperature to 95 degrees, separate it. This is called naturation of DNA. Add primers, bring the temperature down to 37 degrees so that the primers bind to the complementary surface, to the complementary sequence, and again you allow the RNA polymerase to do its job. So now you have from 4 double stranded DNA molecules. So there's an exponential increase in the growth of this particular DNA. In the due course of time, is after about 3 or 4 cycles, is not required, because the double-stranded DNA apparently does not require primer after the 6th or 7th cycles. Okay, so now having understood that you just need a small tiny sample or a few fragments of DNA in order to make such huge number of copies of it, the use of it is very obvious. So what you can do with PCR is you can selectively isolate a DNA sample, and to study them you can amplify and quantify this DNA. You can also study gene mutation. You can take a sample of DNA and make enough copies of it, and compare these copies with various control groups to study genetic mutation. Of course, it can be used to diagnose various disease, where you again take a small tiny sample from either amniotic fluid, and you make multiple copies of it and study the genetic differences or variations or mutations. It's very interestingly used in various medical legal cases and parallel diagnosis. I'll come back to this a little later. And of course, it is used to study cancer. To study cancer, in the sense you can study the amount of mutated genes present in a given sample. So that was about PCR. Now let's move on to gene sequence and karyotyping. I won't go much into this because we've just got ourselves introduced about karyotyping in the previous class. I spoke about spectral. There's nothing much to add to this particular slide, which I'm reusing it. Basically, what this particular technique does is it takes dyes of various different types of various different mixtures, and these dyes are probe enabled. These are radio enabled nucleotides. That means to say you add a certain radio isotope to a nucleotide sequence. It can be carbon isotope, it can be phosphorus isotope, which emits radiation. So you have different radiations being emitted, producing different colors. So you take these radio labeled isotopes radio isotope labeled bases, and you allow them to bind with the chromosome or the DNA of interest, and you get a various visual representation of chromosomes on karyotyping. All right, we will not go much into the technique of this. We'll move on to blotting technique. Again, probes and use of this radio label will be reflected over here in blotting techniques. Blotting technique also is a kind of a continuation of PCR in certain cases. So basically, what is blotting technique? What this does is this technique allows us to identify a given set of desired DNA and RNA using probes, and allowing us to immobilize that particular nucleotide sequence on a solid support. So you identify it and you separate that particular DNA sequence on a solid support, and you immobilize it, you fix it over there. Once you do that, you can extract that particular set of DNA or RNA or protein, and you can further study it. Now there are different types of blotting techniques. One is called southern blotting, northern blotting, western blotting, and southern western blotting. There's another technique called dot blotting as well. Basically, this idea was presented by Ed Southern way back in 1975, where he used this blotting technique to isolate DNA. Because of his name Southern, the technique was labeled as southern blotting. But consequentially, the same technique was used to isolate RNAs, and it was called northern blotting. I don't know for what good reasons. And then protein was isolated using the same technique. It's called western blotting. And then there was another DNA binding protein that was isolated. It's called southern western blotting for the obvious reason. Now, southern blotting technique undergoes this for the following stages. Now, basically what you do is you take the nucleotide sequence of a mixture of nucleotide sequence and you allow them to separate on a gel. Once you separate this particular technique on a gel, now the technique which you can do that is, there are many techniques you can do it. One is electrophoretically you can do it. Electrophoresis means you apply the gel containing a mixture and allow electricity or current to pass through the gel. Now, depending upon the size of the fragment and the polarity of the fragment moves along the electrical grid. And once it moves along the electrical grid, they are separated. The smaller the size is, the more faster it moves. The more charge it is, the more faster it moves. So based on the charge and the size, the DNA fragments move along the electrical grid and they are separated. Once they are separated, the sample is immobilized. Now, immobilization of this nucleoside sequence is done by this blotting technique. What you do is you take this particular gel on which the entire fragment is separated into bands, and you apply a paper, a blotting paper, to take this particular separated genome to transfer the genome onto the blotting paper. So this is a process of immobilizing of nucleic acid technique. Once this immobilization is done, then we hybridize it with probes. Again, as we've spoken about probes in the previous slides, these are genetically labeled probes. You use these probes to bind to this particular fragments. So any particular probe you use will bind to a specific DNA sequence on this separated DNA bands and not to any other. So only as you take these probes and you allow it to react or bind with the DNA samples. Once you allow the DNA to bind, you allow something called stringent wash. You wash away the probes which have not been fixed. So all the unwanted probes are removed. Once the probes are removed, you allow it to be radiographed, and on the radiograph you will know where or which band has this probe bound to. So the probe that contains the radio-labeled isotope of a particular DNA sample of a particular known DNA sequence is binding to the DNA from an unknown sample. So that's basically what this technique involves. Now, as we've learned before, restriction endonuclease is used to produce different cuts of the DNA to produce different restriction fragments, and this particular fragment is allowed to run on a gel, as I mentioned, and the probes are used to identify various sequence of defined sequence of this restriction fragment. This particular video, which was which I will be attaching along with the study material, can help you to understand the entire process in a much more detailed manner or visual manner. Now, basically what you're looking at is the auto-radiographed print. Now, what it is the probes which are fixed onto different fragments. When they are radiographed, they appear like this, and the bands indicate different probes that is fixed. So you study this is the radiographic print of the genetic genome which has been separated. Now, this particular southern blotting technique is used basically for gene analysis. You want to know if you have a defective gene or sickle cell gene. What you do is you separate your gene using RFLP and then your electrophoresis or your chromatography techniques. Once you have this RFLP being separated by these two techniques, you add the probe which is fixed to a complementary nucleotide sequence, and this probe goes and binds exactly at that genome complementary to this particular probe containing nucleotides. And if the patient has got sickle cell disease, this probe will bind to that particular sequence with a defective gene. And when you radiograph it and when you see the particular probe being fixed onto that particular small sequence, you know that this particular individual is carrying a defective gene. So that's genetic analysis. Forensic application, well, restriction fragment which is produced, which produces different sequence, also is used is used in forensic. I'll show you an example of it in the later slides. You can identify an unknown sample to a given individual by using these techniques. Let's say, for example, over here, you have a crime scene. You have a small sample of a hair or a dead skin or saliva or some kind of a sample. Now that sample has got DNA in it. So what you do is you take the DNA, you cut the DNA into various sequence, and allow the DNA to be separated over on the gel using HPLC, that high performance liquid chromatography, or by your electrophoresis. Once you separate it out, you fix it onto a paper, blotting paper, as we've seen in the video on blotting techniques. Once it is fixed onto the blotting paper, you allow different probes. You take a specific collection of probes and you allow the probes to bind to different known sequence. Let's say you take a probe for insulin gene, or you take the probe for corticosteroid gene, or you take a probe for albumin gene. Randomly you can take a couple of probes and you allow the probes to bind to that particular sample sequence obtained from a sample of the crime scene. And now you have three suspects. The three suspects, you take the DNA sample of the same three suspects, allow the same restriction endonuclease to do the same cuts, and allow the sample to be separated again, and allow the same set of probes that you used for analyzing the crime scene. Once you do that, the probes should be fixed at the same spot. Now if you look at suspect 1 and suspect 3, the spots that these probes have fixed for the same probes are at different levels, because each of these individuals have got different kind of a genome, hence different lengths of restriction fragments are cut. So the restriction fragments cut differently in different individuals. So this is your genetic fingerprinting. Each individual have got different sequences in your genome, hence the restriction fragment endonuclease cuts the restriction site at various different lengths, which is not identically matched from one individual to another. So in this scenario, the suspect 2's restriction fragment nuclease matches with the sample obtained from the crime scene, and hence the identity of an individual in the crime scene has been established. There's another example of this can be used, the blotting technique and the entire RFLP and probes. Now, in case of parental dispute, let's say there's a child and the child comes in and says that he is my father and she is my mother, and you have to prove the identity of the individual. Now you do the same thing. You take the sample of all the three individuals, allow the three individual samples to be separated by the same restriction fragment nuclease. You take probes of a certain known sequence of DNA and you allow the probe to bind on the three samples, the same probes. You wash the sample off and then you allow it to be radiographed. Now this is a very good an actual radiographed image of a mother, child and the alleged father. Now if you look at this picture, the mother and the child have got the same genome sequence at this particular probe. So this probe has got the same genome sequence. All right. Now the mother, the child and the father have got the sequence over here. The mother and the child have got a sequence over here. Now the child and the father have got a sequence over here. Now in this picture you have the mother and the child sequence over here and the father and the child sequence over here. Now what this indicates is that the child has to obtain genome either from the mother or from the father. There cannot be any other source. The genome on the child should either come from the mother or from the father. Now in this case, this particular genome of a child has come, one of the genome is come from the mother, another one has come from the father. Similarly, another genome has come from mother, another from the father. Similarly, this particular probe has got from one from the mother and one from the father. Another probe is one from the mother, another one from the father. So all the genome on the child is either from the mother or from the alleged father. So in this case, the father is the alleged father is the father for the child in question. So you can establish the identity of parental in parental disputes. So that comes to the end of all the three techniques, which we have, all the four techniques in fact, which we have looked into, and I assume that we have I've tried to impress upon the basic principles, again nothing in detail, but the overall idea and the impression of what these techniques are. You should be able to probably write a short paragraph of, or maybe one or two paragraphs on explaining the principles, the working principles of this particular techniques. You should be able to you should be able to understand what these different techniques do, or the intention of use of this technique is, and the application, where do you use them. So you should also you should have at the end of this particular lecture, I hope I have expressed or explained importance of how these techniques can be used in medical practice. Okay, with that, I think I'll conclude my presentation here. I strongly encourage you to see the videos that has been attached along with this particular slides, and there are a few practice questions that have been given to test your understanding on this particular lecture. Now, anytime if you this particular lecture, I would like to hear the feedback, both for my analysis and for my and further to improve my presentation, or to improve the material, study material. Your feedback is very, very important. Please give me your feedback to provide to teach you better and to deliver the content even more effectively. Thank you, and see you in the next class.